Hi, this is Amy Proal with the PolyBio podcast, and my guest today is Dr. John Wherry. He's an immunologist and the Richard and Barbara Schiffrin President's Distinguished Professor and Department Chair of Systems Pharmacology and Translational Therapeutics at the University of Pennsylvania. He's also Director of the Penn Institute for Immunology. For decades, John has studied T-cell activity in the context of chronic viral infection. For example, his lab delineates mechanisms by which chronic viral infection can lead to T-cell exhaustion. And lately, John's lab has been studying how T-cells respond to the SARS-CoV-2 virus and vaccines that contain the SARS-CoV-2 spike protein. He's now moving his research into LongCovid, where he's working to determine if the activity of T-cells can be used as biosensors of SARS-CoV-2 persistence in patients with the condition. And with that, John, thank you so much for joining me.
Thanks, Amy. Glad to be here.
Great. Well, I mentioned that you study and have for a long time chronic viral infection and the activity of T cells. Can you tell me about that generally? How do those two topics intertwine?
Sure. Yeah, so this is something that we've been working on for a long time. It's fascinated many people in the field. In general, for the immune response, T cells are one of our two major arms of the adaptive immune response. We have B cells that make antibodies, and those antibodies are important for many things, one of which is to prevent initial viral infections. And they're pretty good, but as we've learned over the last couple of years, they're not perfect. And in many cases, viruses infect us, get through the antibody sort of shield, and then we need T cells. And T cells are important because they can detect the virus where antibodies can't. They can see viruses inside cells where antibodies can't see them. T cells mount an initial response. They very often help clear an acute infection. And what I mean by that is in the first week or two of an infection, you mount a T cell response. They detect virus in infected cells. The T cells, together with the antibodies and other things, completely eliminate that virus from your cells, and the virus goes away. Your T cells have expanded. They've clonally expanded, so there are millions upon millions of them that can recognize the virus goes away, and then you don't need most of those T cells, so many of them die, and the few remaining cells actually form memory, and this memory is the basis for protection long-term, immunological memory and cells that persist. But there's another outcome. There are actually two other outcomes, right? In the case I just described, everything works well, the virus is eliminated, the host returns to health, and everything's great. A second outcome is that doesn't happen and the host dies. Of course, that's not what we want. But there's a third outcome, and that's sort of a stalemate. And that's a situation where the virus actually is mounting, I'll call it counterattacks against this immune response. And at some point, at around the 2-week mark in both animal models and in humans, it seems that the immune system has an evolutionary choice to make. If it keeps mounting this really aggressive response, killing infected cells, you're going to end up damaging tissue so much that the host dies, not because of the virus, but because of the response to the virus. The other outcome, or the other possibility, and sort of evolutionarily selected, is that the immune system decides to stand down, at least partially, and go into a state of deciding that this is going to be a long-term battle. We need to figure out how to contain this invader, even if we can't fully eliminate it. And T-cell exhaustion is one strategy that allows that to happen. The T-cells reduce their activity. They stop their all-out war on the tissues, but they maintain some ability to constantly monitor, basically form a stalemate with constantly pushing, especially herpes-type viruses or other sorts of persisting viruses, into a state of containment. That is good enough from an evolutionary standpoint to get us through reproductive years. But of course, post-evolution, post-reproduction, we see lots of problems with these chronic infections. Now, what's happened over the years is that biological outcome of the immune system seems to be exploited by many persisting infections to allow persistence, to allow transmission, and has been exploited by developing tumors as essentially a place in the ecology of the host that the tumors can actually evade the immune response and kind of hide, slowly growing in an indolent form until they become clinically relevant.
Yes, exactly. So overall, we have this issue where if the patient, let's say, doesn't fully clear a virus, and some viruses that you study are by definition persistent viruses, like the herpes viruses, you have the immune system now figuring out how to manage that, with both the immune system trying to handle the chronic virus and not completely burn out or kill the host, and the virus using its own mechanisms to try to persist. And there's a kind of, as you say, stalemate seems the perfect word to me to describe this moving forward. Now, in the study of COVID or SARS-CoV-2 and long COVID, how are you iterating this understanding into an understanding of T cell activity with SARS-CoV-2 and even into long COVID?
Yeah, so the long COVID question is really interesting. Let's start at the beginning, though, where we have acute COVID infection. There's been a lot of sort of noise around the idea of exhaustion in COVID, both in COVID infection and also more recently, actually, in repetitive vaccination. The data that's out there really does not support the idea that T cell exhaustion, it does not occur following vaccination, despite some papers on that. It has been implicated in the early phase of acute COVID. What we know is that a lot of features of exhaustion, a lot of activation markers or inhibitory receptors associated with exhaustion come up whenever a T cell is activated. And so we know in even severe acute infection that T cells express a lot of these molecules that are associated with exhaustion, like PD-1 and inhibitory receptor or some others. But most of the evidence suggests that that's just a somewhat predictable activation response to a viral infection. Now, some of those patients end up with long-term symptoms, long COVID. And as you get to long COVID and transition from the acute infection to this sort of long indolent phase of symptomatology, let's call it, there are a couple hypotheses for what might be happening. And there are more than this, but at least two of the major ones that have emerged are that there's some induced or sort of revealed autoimmunity, and you have self-reactivity that's driving some of the symptomatology we see in the central nervous system or in other organs. A second, not mutually exclusive possibility is that the virus actually persists in some reservoirs that are hard for the immune system to clear, but persists in a form that leads to persisting activation of the immune response, which leads to something like a chronic inflammatory disease, but where the drivers of that inflammatory signal are really difficult to pinpoint. In that second setting, what you would expect to see is that the T cells that are virus specific would behave very differently than the T cells that have been involved in a response where the virus is completely eliminated and there's no viral antigen left to stimulate the immune system. We know that T cells that get periodic, continuous, or long-term stimulation through their T cell receptor undergo a pattern of differentiation that's really clearly different from T cells that see their antigen stimulation for a week or two and then never see it again. So in long COVID, we sort of predict that T cells might give us a window into this biology and act like extremely sensitive monitors of whether there's continued SARS-CoV-2 antigen expression in the host. And so we're hoping to use that idea to ask about the difference in T cells in people that have had acute infection and then get long COVID versus have had acute infection and then return to completely normal health.
John, yes, that makes a huge amount of sense. And it's really been interesting for me to follow your work, because I think to give people some context, when we're trying to understand with that, the second hypothesis you talked about, that long COVID patients may not fully clear the SARS-CoV-2 virus and it could persist in a reservoir, likely in tissue where it's hard to target, it's low level of the virus. A common instinct for people is to do antibody testing to try to see if people might have the virus. But antibodies, when you measure in blood, that could have been the timeline on which that antibody was created is hard to know. Maybe the person has cleared the virus, but the antibody remains. But T cells have so much more, the lifespan of a T cell is shorter, right? Can you explain this a little bit more? In other words, T cells to me seem like very good indicators of when a recent or persistent infection, because they have shorter lifespans. And if they're still responding to antigen, if you can still tell that they are by understanding their activity, then you can better understand that someone seems like they're infected and there's still antigen at that time.
Yeah, that's exactly right. So we hear a lot about, loss of antibodies over time, especially right now in the pandemic with loss of protective immunity following vaccination. And that's a little bit of a misnomer. what we see actually following vaccination is the very high levels of antibodies that seem to protect us from sort of mild symptomatic infection. They peak after vaccination and then they decline, but they don't entirely disappear. Actually, antibodies have a very long half-life. The half-life of an antibody in the serum is about 21 days, but antibodies get recycled and put back into the serum. So following SARS-CoV-2 infection, you can find antibodies for quite literally years in the blood. Now, they may not be at a high enough level to protect you from getting the next variant infection, but in terms of detecting them to ask when the antigen was present, it's a very, very blunt tool. Now, you can make the analogy to T cells. T cells also persist and they're there for a long time. But the difference is when you look at an antibody, it's sort of like getting kind of a one-page fax on a fax machine. And you get it, it comes out, it's there, you can read it, it tells you something. Whereas if you look at the T cells, they're like somebody dropping off a 1 terabyte hard drive. They not only tell you something by their presence or absence, you can actually open up that hard drive and read what that T cell saw recently. And what I mean by that is if a T cell saw a virus 2 years ago, even just its phenotype of proteins expressed on the surface is going to tell you that this is a T cell. Yes, that virus was there that T cell was specific for, but it hasn't seen that virus in a long time. We may not know whether it's 2 years or 10 years or 3 months, but we know it's not seen that virus in the past week or two. Another T cell, and let's just use as an example a herpes virus here because we have a lot of experience with this, specific for Epstein-Barr virus or cytomegalovirus, they will look extremely different because they're interacting with those viruses that our immune system is keeping in check on a periodic basis. You can even tell the difference. Herpes viruses are fascinating because their genome has basically 3 sets of genes. They have early genes, intermediate genes, and late genes. And that's important because these herpes viruses in this host pathogen equilibrium that's evolved over hundreds of millions of years, for herpes viruses that are in latency, they express some of their early genes. And those early genes sort of go on and off as the virus tests the environment to see if it should reactivate and come out of latency. But they very rarely express these very late genes because they're the genes that are required to actually put the virus together. They're the building blocks of a new viral particle. So for Epstein-Barr viruses, we call these sort of lytic and latent proteins or genes. The T cells specific for lytic and latent proteins are even different in the information they contain, telling us about the amount of antigen, or probably more precisely, the pattern of antigen availability to stimulate those T cells. So the idea here is that antibodies are really important to tell us whether or not, like plus or minus, have you been infected? They might give us some really vague idea of how long ago, based on antibody isotype or decay rates that we might be able to infer. But the hard drive contained within the T cell, both in its surface phenotype and maybe even in the genes it's expressing and other features, gives us orders of magnitude more information to ask this question.
Yes, that's a great explanation. And I also think, to clarify, that you're doing thus far work on, you're going to gather blood samples from patients with long COVID to do this T cell work. But one benefit of that, I think, is that the T cells do traffic through tissue, correct? So if the virus is in a tissue reservoir, then they get that information. They can potentially respond to the viral antigen that's still there in the tissue and then bring that information back into the blood. Is that correct?
Yeah, that's right, Amy. I think that that's one of the advantages. The immune system, almost every nucleated cell in your body interacts with the immune system some of the time, or perhaps on some frequent basis. And part of that are immune cells that are resident in our tissues. But some of that is through these, I guess, itinerant cells that are sort of migrating around the body, sort of surveying for things. And really, the immune system has a fascinatingly efficient system of having information drain from tissues to lymph nodes, where you organize stuff draining out of tissues, especially inflamed tissues, coming into a lymph node is exactly the place that you have lymphocytes flowing through to see what's going on in the tissue. So there's a very, very orchestrated way that the immune system surveys the tissues. And then those lymphocytes, especially the T cells, have to go back out in the blood and migrate around to find the site of inflammation. They also leak out of inflamed tissues. And so the blood is a big bag of cells, some of which are carrying important information. Our job and our challenge is to think about the blood as, really kind of a giant haystack. And there's a needle in there somewhere, and we have to know what it looks like in order to find the information it might contain. In this case, that job is not terribly hard. It may mean that we're searching for some rare cells. We may need large blood volumes, or we may need to actually have a tissue in some case to know what the signal should look like in the blood. But with the understanding of what epitopes from this virus T cells recognize, we kind of have a giant magnet, almost literally, to pull out the cells that are specific for the virus and then interrogate what information they have. And it is literally a magnet. When we purify these cells, we basically use a magnet to push them into one bucket and let everything else go into the other bucket.
That's really cool. Yeah. I mean, and then I guess from that, it does seem, and I understand this does involve a lot of work, and we're not totally at that stage yet, that you could potentially create a test then that might be iterated into the clinic, at least at some point, that could capture T cell activity in the blood of patients with long COVID or other chronic conditions tied to potentially or persistent viral infection. Does that seem feasible?
Yeah, I think one of our one of our goals as immunologists and our reason to be is to make immunology therapeutically useful. And we've made tremendous discoveries, and we've had what I would call maybe three major revolutions in human immunology. The first, of course, was in the early 1980s when HIV hit, and it forced us to understand how the human immune system really worked, because it was where the virus was actually killing people. The second maybe is the cancer immunotherapy revolution, where we've finally harnessed the power of the human immune system to cure disease, and cure disease that was completely intractable before. But the third revolution has been the last 2.5 years of the pandemic, where as good as we felt about ourselves after cancer immunotherapy of the 2010s, we were humbled in a very short time with the recognition that we don't understand enough. The promise of these three revolutions is that we should be able to get immunology into the clinic in an actionable way. T cell tests in the clinic are very difficult. They're cumbersome. They're variable from the standpoint of getting them approved. Their variance is large, and they're very sophisticated tests from the standpoint of getting them to perform exactly the same way every single day of the year. Vaccination for SARS-CoV-2 has raised the specter, or provoked a lot of us to argue that we need T cell tests as a readout for many of our vaccine studies. Antibodies are insufficient to really tell us the whole story, and T cell testing may add to that story. There are at least a couple of T cell based tests that have been moved into the clinic during the pandemic. There was one that was actually available before that to look at tuberculosis specific T cells, and there are variants of this that have been used for SARS-CoV-2. We have the foundation to think about clinical testing for T cells. That will be extremely helpful for long COVID, but we need to actually take, I think, one more step forward. We don't need to read that entire 1 terabyte hard drive, but we need to actually get a little more information than just are T cells present or are they not present. And it should be possible to do this, but I think one of our major efforts that we should think about, in addition to the really important mechanistic science on how T cells work, what is T cell exhaustion, what are these patterns of activation of T cells in long COVID, some of our efforts should be, great, you've shown that you can do it on a Tuesday. Show me that you can do that every day of the year with high confidence, and you can make it actionable in the clinic, right, so that we can put that back in the electronic medical record as something that we can use to help long COVID patients.
Definitely. Yes. Those are all really good points. I do think that overall, one takeaway from this is that it seems obvious, I think, to us, but that the average clinician right now, when a long COVID patient comes into the clinic and tries to see a doctor about their symptoms, cannot really do a test that would show much about the patient's T cells. What we've talked about thus far in this interview, what you've discussed, cannot be captured in a clinical test too much at all. And so that is part of the reason why a doctor will sometimes run blood work on a long COVID patient and say there's nothing wrong with you. But really, an understanding needs to be among clinicians more that there are limits, great limits thus far, in what can be measured in a patient with long COVID to really understand these really important nuances associated with what is happening.
No, that's so correct. And, you know, our long COVID physicians are heroes, and what they've managed to do in a relatively short time is nothing short of amazing in building, you know, questionnaires and criteria to really start to understand the heterogeneity of this disease and the features of it that are important to recognize. But we haven't given them the tools to do what you just said. And I'll give you sort of two thoughts on this that crystallize this. If you go to your physician, I'll call him a physician, whether there is no clinical immunologist to go to ask this question. We have allergy and immunology in the clinic, but if you go and ask your long COVID physician, how healthy is my immune system? We use tests that were to be like the most kind, developed in the 1970s. You know, pretty much you're going to first do a white blood cell count, which frankly is about 150 years old. So we need to do better than that. If you take that same blood sample and, you know, now invented in the 1970s and early 1980s, you can do a differential. So you can know, you can count how many T cells there are versus non-T cells, just total T cells. You know, you're basically looking at the entire haystack. You take that same blood back to any number of research labs that are working in these areas, and in an afternoon, you can have several 100 million pieces of information about the immune system. The gap between what we can do clinically and what we can do on the research side is just absolutely enormous. This is not because we don't want to get that sophisticated information into the clinic. It's because we don't know what to do with it when we get it there. So it's a two-part problem. If we knew we could make a decision with that patient sitting in front of you today, that having those millions of pieces of information, we would give them a different treatment or a different drug, we would develop the way to get the test rigorous. We don't know what to do with the information yet. And so I would encourage long COVID patients not to fault their physicians when they say, you look healthy, we don't know what we can tell you. There's a gap elsewhere, and part of that gap is in the basic knowledge of how this biology has gone awry, and we need to really get better information about that, and then make it actionable in the clinic.
Absolutely, and with the long COVID physicians, they would say, you know what, there's only so much we can measure right at this point in time. So we validate you certainly, but there's limits to what we can do, as opposed to saying, for example, that the condition is psychosomatic or something like that, which can happen. So hopefully that would not occur. But yeah, well, John, then I'm interested, the T cell immunotherapies then, I mean, looking forward, we do know, as you explained before, that second revolution where immunotherapies for T cells were brought into cancer, and viral pathogens are connected to many cancers. Do you see, again, down the road, any efforts then to bring T cell immunotherapy into COVID or long COVID? I know it's pending still data and more research, but do you see that as a possibility down the road?
Absolutely. And I think that I'll tell you, you know, some pieces of data I think we're going to get that will be very useful to further answer that question. And then some of the things we could think about, we have some things on the shelf that maybe actually could be very interesting to use. So there are vaccine efforts going on in long COVID patients, right? Yep. So if we boost the T cell response, does that change the trajectory of disease? And, you know, early in the recognition of long COVID, there were a number of observational reports about vaccination helping or making or exacerbating symptoms of long COVID. And looking at that data, I think it broke down more often that there would be a benefit, but it certainly wasn't entirely clear. Sometimes symptoms were exacerbated. And that may clarify as we look at the heterogeneity of long COVID and recognize that it's not one thing. And that's going to be very important and helpful there. So that's going to be a huge, important set of information when we vaccinate long COVID patients. A, what happens to symptomatology and disease? And B, what do their T cells tell us? The T cells, again, are going to be carrying information with them. If those T cells have not seen virus for months to years, there's going to be a characteristic type response, actually quite similar to what we're seeing with probably breakthrough infection or booster vaccines, where you see T cells mount a really good dynamic recall response. If, however, those T cells have been chronically stimulated for a long time, we're going to see quite a different pattern. And so, in other words, the boost of a vaccine in a long COVID patient is going to be less effective, not less effective to prevent disease, new infections, but it's going to be less effective at actually getting a really robust T cell response because the T cells are going to be maybe exhausted. There's a little bit of evidence for that, although I think it's very early days. Maybe they're just going to have been stimulated so much that they kind of make a weaker response. We see this for CMV specific cells, that they're, I'll call it a bit sluggish to be sort of generic about what we know about CMV specific cells. But once we know that, like, are the T cells involved here? Cancer immunotherapy and a little bit of HIV work has given us a treasure chest of T cell based therapeutics that can be used. We have checkpoint blockade, anti-CTLA-4, anti-PD-1 that reinvigorates the immune response in cancer patients. For anti-PD-1, mostly by targeting exhausted T cells. So if we find exhaustion in long COVID patients, we already have a drug that counteracts exhaustion, at least temporarily. If we find that we just don't have very good T cell responses getting primed, we have things like CTLA-4, we have other cytokines that are now being used as therapies in cancer patients. We have a wealth of things on the shelf from the cancer world that could start to be applied in long COVID. We also know that sometimes we just need to redirect the immune response. It's not that the immune response isn't there or isn't working right, but it's gone down, the left fork in the road versus the right fork in the road, and we need to actually sort of back it up and go down the other path. We have a lot of immune redirecting type of therapies now too. Sort of anti-cytokine antibodies are probably the most clear, but there are other ways you could sort of take the immune response back from the wrong kind of inflammatory response and redirect it. And there may be hints of that in long COVID as well. So I think there are a variety of options, and we could think about which of those might be worth trying in long COVID. We need to recognize that it's more complicated. There is no free lunch, especially in the immune system. If you push the immune system to become more activated, you run the risk of things like autoimmunity or provoking inflammatory diseases. So we're going to have to be very thoughtful about this, especially if that other hypothesis about long COVID, about autoimmunity, is on the table. Like, we may be dancing on a knife's edge here and want to be very careful about where we apply these sort of T cell activating therapies.
Yes, that makes a huge amount of sense. And I think that, for example, I know that with the cancer immunotherapies, where T cells in simple terms are the activities energized again to help the T cells attack the tumor, there is what's often called immunopathology, whereas the T cell now with its reinvigorated state attacks the tumor, there is a rise in the patient's symptoms because there's more of this, a battle, should we say, that's occurring between that, and that leads to a rise in symptoms. But then there are better outcomes in patients who are sort of able to manage those symptoms with a physician. The long-term outcomes improve. So does it seem that there might need to be some immunopathology? I mean, this is definitely looking ahead, but and also the other thing to say is that it just seems clear to me that this is going to have to be personalized medicine at a certain point with these patients. You've just given all these different instances. Well, someone might have more. Again, we're talking about, as you made clear, it's very unlikely that every long COVID patient has the same exact thing happening to them. So in other words, it seems that, like what you said, with being able to measure T cells, which is why research is still so important to be able to have the diagnostics on this, will also help inform the possibility that treatment even may differ from patient to patient. But going back to the immunopathology, do you think might be that paradigm might end up coming back into going beyond cancer?
Yeah, I mean, the immune system does make us feel sick. I mean, this is, when you have a severe viral infection, yes, the virus could cause some pathology, but it's typically your innate immune system and then your adaptive immune system that makes you feel sick. Sometimes, as we talked about with acute COVID, it's actually that immune response that ends up killing you, and the wrong time or too late, and you end up with an irresistible force hitting an immovable object, right? And that's a recipe for disaster. But a little bit of symptomatology with T-cell mediated therapies is often a good prognostic indicator. You're playing with fire though, and we have to be thoughtful about this. And the very classic story that's been told many, many times is in one of the earliest CAR T-cell patients, treated with engineered cellular therapy for acute lymphoblastic leukemia. Emily Whitehead, was treated with CAR T cells, and about a week later spiked an incredibly high fever, and the CAR T cells were actually killing her until Carl June and Steve Grupp and others recognized the opportunity to actually treat some of those inflammatory symptoms, a cytokine called IL-6, and block it, that you could curb the really dangerous inflammation, get through that dangerous phase, and actually she remains cancer-free 8 years later, I think eight or 9 years later now, something like that. So I think the lesson there is not just that sometimes the immune system causes damage. It does a lot of the time, and that's how it eliminates things that are hard to eliminate, but also that we need a really good knowledge of what to expect. Now, let's take 2 examples of this. If we have a major hypothesis that the virus, that SARS-CoV-2 is persisting, let's say still in pockets in the lungs and in some lymphoid tissue, maybe in the gut, and there's some evidence for that. We know where to look. We can characterize some symptoms we might expect: pneumonitis, perhaps IBD-like intestinal inflammation, and we can anticipate how to treat those things and get patients through that window of perhaps increased symptoms. If, however, most of the virus is in the central nervous system, we have a slightly different challenge on our hands. It may not be insurmountable, but we need to have a little bit better understanding of that, or we need to be very, very careful in the way we think about T-cell activating therapies and trials in the first stages of treating long COVID patients.
Absolutely. Yes. And that's why I think it's so important that I do think, some clinical trials are beginning in long COVID, and that's great. And I think that clinical trials can be very useful in that sense, to be able to understand, how do patients respond to a particular therapeutic, but at the same time, a huge amount of robust research needs to occur at the same time to continually understand exactly all these different nuances that we're talking about, because that's really going to allow us to understand really what's happening and the potential adverse effects and everything, either beneficial or adverse, that we see people report. I just emphasize, we need so much research on this to be able to get to these clinical points.
Yeah, I couldn't agree more. I'm going to pick up on a thread that you brought up a minute ago and add to that comment. And that is that we should learn from every single patient who gets treated, and not doing so. Well, doing so is very expensive, cumbersome, time-consuming, and something that can't be done at every institution. Not doing so is not acceptable. Every patient is telling us something really important, and every patient is unique. You brought up this idea of individualized therapy or personalized therapy, and many diseases have a genetic underpinning or a genetic contribution. We know even in settings in the immune system with autoimmune diseases, where we have good genetics that are known to contribute to disease, that predispose you to disease, 70% of the penetrance of disease is still non-genetic. You can have identical twins with the same variant, and 70% of the variance in whether one or both of them get it is non-genetic. What that means, it doesn't mean genetics doesn't predispose you and play a major role in determining whether you get autoimmunity or not. What it means is that our immune systems accumulate a lifetime of experience that influences every future immune response. So we have to be looking at individuals. We have to be looking for patterns. It's not that we're going to see, every person on earth is a unique situation in all respects. But what we're going to see is that every person has unique patterns that converge on some common themes. But that doesn't mean that all rheumatoid arthritis patients have the same path to get to their disease. And for sure, not all long COVID patients are going to have the same underlying drivers of their long COVID pathology. Even if all of them have viral persistence, the way their immune system is wired to provoke disease manifestations is going to be a little bit different.
Yes, absolutely. Yes. And in other words, genetics, in a sense, set the stage for what the environment then does to a person that gets involved in chronic disease. And with that in mind, where we're talking about what is impacting the immune response over time, we do get to this second issue in chronic disease generally tied to infection, but also long COVID. Another trend in long COVID is that some teams are finding that the herpes viruses, including Epstein-Barr virus, seem to reactivate more often, give or take, in patients with long COVID. So now we have this interesting phenomenon where you could have a patient that doesn't fully clear the SARS-CoV-2 virus. I don't know, but perhaps Epstein-Barr virus reactivates in the patient too. So we have now different viral viruses in a case. How, it's probably, without making it too complicated, how did these different viruses affect the immune response in T cells? Like, how can this be pulled together into what ends up being the full disease that someone might have?
Yeah, this is, I think, a really important question. I mean, these are fascinating initial observations. We need a lot more information to really understand what's happening here. But the data on a connection to Epstein-Barr virus looks really interesting. And there's other independent studies on a potential link between Epstein-Barr virus and multiple sclerosis that have nothing to do with SARS-CoV-2. So, there are a few things pointing us at this virus that we thought was kind of an innocent freeloader in our immune system that may mean that it's actually a little bit more of a bad actor than we previously thought. We don't know how this would work for SARS-CoV-2. I'll give you an example of a situation that we know from a completely different setting that highlights this idea of the sort of hit and run that can provoke disease. And you end up kind of focused on the wrong thing because you're looking at a time when the person who threw the match on the tinder has long been gone, and you're focused on the tinder, not the person who started. And that's an example of a series of studies that a person named Ken Cadwell did when he was a trainee with Schiffrin at Washington University in St. Louis. They were looking at the development of intestinal inflammatory bowel disease in the intestines of humans. It was known that there was genetic association. There's a variant of a gene called ATG16L1. Doesn't really matter what it is. It's a gene that's involved in regulating cell biology, especially of cells that have to do a lot of secretion of things. That was known to be a risk factor, but it was entirely unclear how that was linked to development of disease. Why did some people with that variant get disease and others didn't? So what Ken showed really beautifully was that there's a virus called norovirus. You may have heard this is sort of the cruise ship diarrhea virus. It is a virus that causes diarrhea from time to time, especially in confined living quarters, cruise ships, nursing home facilities, and other inpatient facilities. It's pretty inconsequential most of the time, unless you're very old. Kind of isn't a fun thing for a couple of days, but you recover and it's no problem. Unless you have a variant of ATG16L1, in which case you have a high risk of actually of converting to inflammatory bowel disease. But the connection to this norovirus was completely unknown for a long period of time. Because what Ken showed is when you take mice that have the same mutation as humans and give them norovirus, they come down with inflammatory bowel disease. But by the time they have symptoms, the virus has long been cleared by the immune system and is nowhere to be found. But actually now, because you've disrupted the function of these cells that need this protein, the microbiome, the normal component of our gut, now becomes the inflammatory signal that keeps driving this inflammation. So it looked for many years like the microbiome was the problem. But it was actually this distorted interaction between the microbiome and the host, because of this background susceptibility, that was causing the disease propagation, whereas the norovirus was the original cause. So that relationship, exactly the way it turned out there, is not going to be what it is between SARS-CoV-2, Epstein-Barr virus, and long COVID. But the concept that SARS-CoV-2 may be provoking a change in the way Epstein-Barr virus is managed by our immune system, leading to now a cascade of events that would never have occurred if it weren't for SARS-CoV-2, but where Epstein-Barr virus looks like the smoking gun, could be a big part of what's happening. Now, we had more information in the example I told you from Ken's studies. We don't yet understand the context of EBV here, and we don't have a good mouse model that's exactly like EBV and SARS-CoV-2. So it's going to take some work to really dissect that, and we may be able to do more of that in humans now than we could have many years ago. But I think it's going to be a really, really important thing to dissect. We should remember that Epstein-Barr virus is not the only one of these persisting herpes viruses in humans as well.
Exactly, right. And I mean, it's just very interesting to think that as humans, we all accumulate over our lifetimes these persistent viral pathogens enough. The herpes viruses, but like you said, Epstein-Barr virus, HHV6, to other degrees, varicella, other herpes viruses, even other parvovirus 19, there are other, and even bacterial pathogens to an extent that can also either act in a more commensal friendly state in simple terms, or under conditions of inflammation or immune dysregulation, change their gene expression to act more as pathogens. For example, or there are just bacterial pathogens, P. gingivalis, bacteria and oral pathogens. These organisms that it's similarly to viruses, and the mechanisms are all different, but they can sort of be contained by the immune system when the person is in a state of balance and the immune system's in decent shape. But then if you lose immune control and in a new infection, like SARS-CoV-2 can do that in a number of ways, these other pathogens or organisms can change their activities. We have this whole second layer of what I think can happen under that also begins to matter in these conditions. And so it's another variable. Well, interesting. John, how did you get interested in this topic in the first place? What made you become a researcher that studied chronic viral infection, T-cell activity?
Yeah, it's a good question. So I think from my beginning in immunology, I was most interested in host pathogen interactions, and this sort of two-way evolutionary battle between microbes and the immune response that contains microbes. And during my PhD, I studied CD8 T-cell responses to viruses. And during my PhD, the very first papers were published on this concept of T-cell exhaustion. Instead of when things go well, we might want it to go better when we cure acute infections and all of that. The bigger problem to me was what happens when that first initial interaction doesn't clear the infection, and you now have this prolonged interaction over time with this constant testing and retesting of whether the virus is winning or whether the immune response is winning. That's a really dynamic battle, and figuring out how that dynamism plays out over time, like, immediately fascinated me. So I decided to do my postdoctoral training in one of the labs that really first described this T-cell exhaustion. And it's become kind of a minor obsession that's grown from just this host pathogen interaction to these exhausted T cells being a microcosm of developmental biology, and has given us a window into all the decisions a cell has to make as it views its environment, assesses what the environment is doing, and then converts those cues into information to decide which fates to take next. We were, I think, as happens in science, I think a little bit lucky that this cell type turned out to be incredibly relevant for chronic infections like hepatitis B and C, HIV, to cancer. Almost all cancers that are highly mutated or can be recognized by the immune system have some component of T-cell exhaustion involved in their pathogenesis. Many of them, exhausted T cells, are the main mechanism of response to checkpoint blockade. And now we know that exhausted T cells are a major feature of autoimmunity. How they exactly participate there, I think, still remains to be seen. I think many people would say that that we want exhaustion in autoimmunity because it's sort of the inverse of cancer and chronic infections. I think that assumes that exhausted T cells are non-functional, which is not right. Exhausted T cells are chronically functional at a low level, not enough to cure chronic infections or cure tumors, but maybe enough to cause tissue disease in autoimmunity, where there's this sort of chronic low-level inflammation. So, I think it's become now this fascination of trying to understand how these cells work, being deep in the weeds of the mechanisms of developmental biology, but also feeling an extremely strong push or responsibility to put our money where our mouth is. And if this biology is important clinically, I sort of feel like we have an obligation to take that next step and go from the things that we can understand in experimental models to saying which of those are actionable and useful in the clinic. So I don't know. I think your career takes twists and turns, and we've sort of been fortunate to be in the right place at the right time a lot, studying this interesting cell type.
Yeah, I mean, you've been fortunate, but if you look through the papers that your lab has done, you've also worked very systematically on these topics. That's really interesting. I read your paper, I think it's in 2011. I think it's something called Understanding Chronic Viral Infection. And that paper had a big impact on me. I was really interested in the topic at the time, and it's what led me to actually pursue my PhD in similar work. And coming from reading that paper where you discuss a lot of the relationship, again, the dynamic relationship between the immune response and what it's doing and the pathogen and what it's doing to the immune response, and this whole relationship between that, but then watching you iterate it over time into more specifics, including, being able to capture what a T cell can do to a specific target antigen, as it travels through tissue. You've done a great job in building slowly forward. And I see you still have a lot of work on your plate to move forward with, plenty of challenges still. With that in mind, with pathogens, persistent pathogens in chronic disease, you mentioned MS, and you mentioned now this recent Epstein-Barr. There were two big studies recently that connected Epstein-Barr virus to MS. There was a study at Harvard and a study at Stanford. The Stanford study was really interesting because mechanism was delineated in that study, where it actually seemed there was cross-reactivity between a protein that Epstein-Barr virus makes in a human protein or with a similar structure. And that cross-reactivity actually is part of the disease process, where the immune system is actually potentially targeting a viral protein and it reacts with a human structure with similar size and shape. That, with these understandings, and that's sometimes called molecular mimicry, how much do you think now, as you said, we know the genetics, there's a human genetic component to most chronic disease. Where do you think we're going to go from here with MS, other so-called purely autoimmune diseases, lupus, rheumatoid arthritis, even? How much do you think these chronic viruses that we haven't really maybe been taking as seriously as we could, how much do you think that's going to grow into a better understanding of these others, potentially diseases that are called purely autoimmune sometimes at this point?
Yeah, I think there's a huge opportunity and responsibility to actually get a better handle on that. We're starting to invest heavily in autoimmune immunology here at Penn, and part of that is going to be to better define the individual's immune landscape in autoimmune diseases that have the same name and same clinical presentation. No offense to the rheumatology world, what's happened in rheumatology, especially over the past 10 years, has been nothing short of remarkable. But many, if not most, of our rheumatological drugs treat symptoms, not causes of disease. And we have seen this over and over again in trials of different diseases, where we give one drug to everybody with a disease with the same clinical name, and the drugs often fail because they don't meet their trial endpoints, and yet there's a small subset of patients that has a remarkable response. And I think when you look across the board, that might just mean people are all different. It might also mean that some people have that target as a symptom of the disease and others have it as a driving cause. I think without knowledge of some parts of immune history, we're going to miss a lot of opportunity to treat patients with drugs that would be better suited to their flavor of that disease compared to patients who have a slightly different flavor of that disease. One example of this, again, from I think the same group at Stanford or related group at Stanford, is if you separate people into those that have human cytomegalovirus and those that don't, and give them the flu vaccine just as like a surrogate readout of immune function, they're different. Having CMV makes you respond to the flu vaccine differently. If it can do it for, no offense, but kind of a crummy vaccine, imagine what it can do to a pretty potent response, or a response that starts to get miswired and has all these positive feedback loops to keep going that way. Now, CMV may not be what underlies autoimmune disease. I'm not making that connection. Or seeing hints that EBV might be in some cases, maybe through molecular mimicry, maybe because it's setting up a different organization of the immune system only in people who have a certain genetic background, or only in people who also have a combination of 3 other persisting microbes just in that combination. So we need to better define individual immune fingerprints as a script for what how the immune system has been programmed in individual people to that point in their life. We should also then be tracking that prospectively so that we can tell when the immune system is starting to give us information about a changing trajectory of health. This now becomes the opportunity to treat people to maintain health rather than be on our heels all the time and treating only when disease manifests. This might be especially important in people that have genetic predisposition for certain diseases, when we can treat them before disease evolves.
Yes. Yes. And by the way, I love that study you mentioned with the CMV, cytomegalovirus, and the flu vaccine. It's Peter Broden, who's now at Karolinska, but was at Stanford at the time of that study, was the lead on that study. And it's funny, on my Twitter, the background image is an image from that paper, because that paper is so important to me. I try to put it on people's radar every time we talk. So I'm glad you brought it up. But everyone should read this paper. I'll put it in the chat of this podcast. But yes, last, aging. So when we hear about aging, and I'm on a couple groups that are looking at the mechanisms of aging. Mm-hmm. How much do we think that what we call healthy aging, or the aging process, is actually very influenced by this? Pathogens that people acquire over time, the immune response, and how it is modulated by these pathogens. How much of how does this contribute potentially to just human aging?
I think probably a lot. I mean, there are a lot of good studies looking at the role of chronic inflammation and aging, and there are very, very strong associations. And there are even some studies that actually have implicated chronic inflammation in a causal way with, aging's, increasing dysfunction of the immune system over time. There's one sort of underpowered study that I'd like to quote, because I think it's probably I'd call it more of an anecdote or an example, but I think it illustrates the story. There's a study, and I couldn't find the reference if you asked me to, but if you look hard enough, you'll find it, of octogenarians, so people in their 80s already. If you then segregate them into those who are CMV positive versus CMV negative, that predicts lifespan. Overall, CMV positivity does not predict lifespan, but if you make it to 80, being CMV positive is a bad thing. Probably because you have a higher level of baseline inflammation. And it's not the inflammation from when you're 82 to 87 that gets you. It's that you've probably accumulated a lifetime of greater inflammation because of CMV, or at least that's the implication of those studies. I have a friend, Stan Plotkin, who has been talking to me for 20 years about making a CMV vaccine. Mm-hmm. CMV is a really important problem in newborns and transplant patients, but his interest is actually in this potentially broader impact on overall health over time. And, I think we should be having more conversations about those kinds of things, about whether we should manage our virome in a way to optimize long-term health.
Yes, absolutely. Because as you said, it seems to me that the dream for medicine would be to treat people before they even develop the problem that they have. And if we know that there are viruses that have such a potential to drive underlying chronic inflammation or chronic disease processes, it becomes a question of if we should try to manage them before they really act up to do that. And so I think that along those lines, thinking about the idea of vaccination for some of these viruses that just infect us often and can do many bad things, even though they can coexist with us, is a very interesting idea.
Treating health is much cheaper than treating disease.
It certainly is. That seems agreed. Well, John, thank you. This has been so interesting. It's always really, truly interesting to speak with you. And I'm really excited for your research, including on LongCovid, to continue to move forward.
Yeah, thanks, Amy. It's been fun.